Hello. Now we'll discuss different experiments that led to discovery of DNA. The first one is bacterial transformation that implicates DNA as the substance of genes. This experiment was carried out by Frederick Griffith in 1928. He used Streptococcus pneumoniae organism, smooth strain of Streptococcus pneumoniae, smooth, that is the one which have capsule around it. Now you know that capsule of bacterial organism is extracellular polysaccharide present around the cell, which makes organism poisonous or virulent, and the rough strain of the same organism which is non-capsulated, which is not virulent, a virulent strain. Okay. Then in 1944, Avery, MacLeod, McCarthy, these three people, they determined that the DNA is transforming material. Now the next is evidence for DNA as genetic material. Griffith in 1928 discovered the process of transformation, that is something is transforming non-virulent strains to virulent one. Avery, MacLeod, McCarthy in 1944 discovered that DNA is transforming principle, and Hershey and Chase in 1952, just before 1 year of the discovery of DNA structure, discovered that the viral DNA is transferred from one generation, or say from parents to the progeny. And next one is Guthrie and Sinsheimer in 1960 stated that viral DNA codes for complete virus, that is, it codes for the proteins which helps in assembly of complete virus particle. Now this is the Griffith experiment. See what he did. He used capsulated, so therefore the smooth strains of Streptococcus pneumoniae, and injected to the mice. Observed that mouse dies. Then he injected rough strains which are non-capsulated and therefore are non-virulent, injected to the mouse. Mouse leaves, healthy mouse. He found that mouse does not die. Then he killed smooth strains, that is virulent form of Streptococcus pneumoniae. He killed them with the heat and used that preparation of dead organisms injected to the mouse. Mouse does not die. And then he used mixture of heat killed smooth bacterium with the live rough cells. Both are mixed and injected to the mouse. Mouse dies. And then he found in the dead mice the strains of smooth Streptococcus pneumoniae. Now how this is possible? Because he used initially dead cells, dead smooth Streptococcus pneumoniae for injection, and rough live strains mixed with dead cells. Then how he acquired these living S forms in organism in this mouse? So he stated that something is transforming this non-virulent strains to the virulent one. Okay. This is the diagram of same experiment which Griffith carried out. Then Avery, MacLeod, McCarthy, what they did is they mixed heat-killed S components in the medium along with the live rough form, incubated for some time. After some time they found that this living non-virulent form, that is rough forms, are being transformed to the smooth form, that is virulent one. Now this is the experiment that showed that the DNA, not the RNA, was transforming principle discovered by Avery, MacLeod and McCarthy. What they did is they took the mixture of the DNA and RNA extracted from the organism. They treated with RNases. Now the RNases is an enzyme which degrades RNA molecule. So what will remain is only DNA. Okay, here only the DNA remains because this mixture is treated with the RNA. So all the RNA molecules, they are denatured, they are degraded. Okay, then they have added this DNA mixture to the rough strains of bacteria. Same organism they have used, Streptococcus pneumoniae. Okay, only the DNA they have added to the rough strains of bacteria and allowed those bacteria to grow onto the agar medium. Okay, later they have found that these colonies which are developed, they are sticky in nature, that is, they are capsulated organisms, they are smooth forms of smooth form is developed. Now how this is possible? Then they stated that the DNA enters into these rough strains of organisms, and these rough strains are then transformed with the smooth one. As the smooth strains organism DNA enters the rough strains, these rough strains are transformed to smooth one. Therefore they discovered that the DNA is transforming principle which is converting one form of organism to the other. Okay. Second, they used the mixture of the DNA and RNA treated with the DNases, where all the DNA molecules are cut off, denatured, only the RNA remains. Then they mixed this RNA preparation of smooth strains of organism with the rough strains and allowed them to grow onto the plate. There is no transformation of this rough form to the smooth form. So this experiment tells us that this is the DNA molecule which transform the organism to the other form and not the RNA molecule. Then third experiment is Hershey and Chase experiment. In 1952, Alfred Hershey and Martha Chase provided convincing evidence that the DNA is genetic material. Okay, they have used bacteriophage T2 for their experiments. This experiment is also known as Blender's experiment. Okay. To determine this, that the DNA is genetic material of an organism, they designed an experiment showing that the only DNA of T2 phage and not the protein enters the E. coli cell and is passed to the progeny of phages. They concluded that the injected DNA of the phage provides the genetic information and is passed on to their progenies. Okay, so this is an experiment. See, the first stage, batch 1, for batch 1 they have used radioactive sulfur, heavy sulfur S35 in phage protein. They have used S35 for labeling the phages. Now why sulfur is labeled? Because there are in proteins there are sulfur containing amino acids present, you might be knowing, cysteine and methionine. So this sulfur is used for labeling of proteins. And heavy phosphorus, see this one is batch 2 where radioactive phosphorus P32 is used for labeling the DNA, since the DNA is composed of sugar, phosphate and nucleic acid. So from the sugar, phosphate and nucleic acid, phosphate is labeled. So this stage 1, batch 1, labeled phages, the phages whose coat, protein coat is labeled is allowed, those phages are allowed to infect to the E. coli cells. Okay, as E. coli cell infect to the phages, they allowed this viruses to multiply inside this E. coli cell. Then they have lysed the cell suspension in blender. Why? Because agitation frees out phage parts from the cell, and whatever is enters phage, say whatever phage parts are entered into the cell, they will remain into the cell only. All the phages which are only present at the surface of the cell, they are removed out. Then they centrifuge the suspension. They have analyzed pellet and the supernatant. Okay, in the pellet they found the E. coli cells infected with the phage, and in supernatant they have found radioactivity. That is, they have found radioactivity because the protein codes of phages are labeled, which does not enter into the cell. In pellet they did not find any radioactivity. In batch 2, phages with the radioactive DNA are allowed to infect to the E. coli cell. Then blender is used for separation of phages. Now here into the cell you can see this blue colored particle which are labeled DNA molecules. These labeled DNA molecules are found into the pellet when this suspension is centrifuged, and no radioactivity is found in supernatant where only phage particles are present. So this dictates that this is the DNA molecule which is transferred into the cell. Okay. The next one is structure of the DNA. Okay. Now we know that the DNA is made up of sugar, nucleoside or nucleic base, and the phosphate group. Okay, these are the DNA constituents. Now if you see the difference between the DNA and RNA, DNA and RNA both are nucleic acid, deoxyribonucleic acid and ribonucleic acid. The difference between the two is very simple, as the name dictates, deoxyribonucleic acid. Into the DNA, the sugar which is present, the ribose sugar, is deoxygenated at carbon number 2. See here, this is deoxygenated carbon. Here in ribose, hydroxyl group is present, OH group is present. Here only hydrogen is present, so it is deoxygenated sugar. Okay, so this is the basic difference in DNA and RNA. In DNA, deoxyribose sugar is present. In RNA, ribose sugar is present. Okay, these are the 5 bases which are present in DNA and RNA molecule. Bases are of two types, purine and pyrimidine. Purines are adenine and guanine. These are double ring structure. Here you can see these are double ring structure with the 9 carbon atoms. And here pyrimidine is single ring structure which includes cytosine, uracil and thymine. Now here if you see this adenine, now see numbering of this purine starts from this carbon number 1 and runs in anti-clockwise direction for this first string, number 1, number 2, number 3, number 4, number 5 and 6, and then it jumps here 7, 8, 9 again in clockwise form. And for this pyrimidine ring, numbering starts from this base nitrogen which is to the downside, number 1, 2, 3, 4, 5 and 6. Now adenine, if you see here at the 6th position, amino group is present. So the purine name of adenine is 6-aminopurine. Guanine is 2-amino 6-oxypyrimidine, because at the second carbon amine group is attached and at the 6th carbon oxygen is present. So guanine is 2-amino 6-oxypyrimidine. Then cytosine is 4-aminopyrimidine, 4-amino 2-oxypyrimidine. Uracil is 4-2,4-dioxypyrimidine. Uracil 2,4-dioxypyrimidine. And thymine is 5-methyl 2,4-dioxypyrimidine. Now you can see here in thymine and uracil only one difference is there. At the 5th carbon in thymine, this 5th carbon is methylated, 5-methyl 2,4-dioxypyrimidine. Okay. This uracil is the base which is present in RNA molecule, not in the DNA. So in DNA, ATGC, adenine, thymine, guanine, cytosine bases are present, and in uracil, AUGC are present, adenine, uracil, guanine and cytosine. Okay.